Welcome to the Explainer. Our bodies have this incredibly highly tuned waste processing system, especially when it comes to our red blood cells. But when that system gets overwhelmed, well, these systemic effects are immediate. Today, we're totally demystifying hyperbilirubinemia. We're going to break down the physiology, the biochemical basis, and look exactly at how red blood cell breakdown happens and what goes wrong when this super-regulated pathway gets disrupted.
Okay, let's dive into this. Here is our roadmap for today's explainer. We've got 5 sections. 1. What is hyperbilirubinemia? 2. The source of bilirubin. 3. The biochemical pathway. 4. Types of hemolysis. And 5. Clinical signs and findings. Let's get to it.
Section 1. What is hyperbilirubinemia? What exactly is it?
So think about this for a second. Why does the breakdown of our own blood cause us to literally turn yellow? When a patient presents with jaundice, that distinct yellowish hue, it's a massive clinical clue. But bridging that gap from normal red blood cell destruction to systemic yellowing, that requires us to look really closely at the breakdown process and the exact molecules left behind in the bloodstream. Let's define it. Hyperbilirubinemia is simply a state of excess bilirubin in the blood. That's the core definition. And while jaundice is the physical sign you actually see, the underlying pathological driver is often hemolysis, the breakdown of red blood cells. So let's journey inward to see the microscopic mechanisms driving this whole state.
Section 2. The source of bilirubin. Where does it actually come from at the end of the cell's life cycle?
Think of the spleen as the body's premier cellular recycling plant. When red blood cells undergo extravascular hemolysis, this is happening mostly right here in the splenic macrophages. Basically, the red blood cell is broken open and hemoglobin is released. Now, the body doesn't waste anything. The globin part of hemoglobin, that's separated and sent straight back to the amino acid pool to build brand new proteins. But that leaves the heme portion behind. And this heme is the raw material that now has to undergo some very specific enzymatic processing. Let's move to section 3 and see how this builds. The biochemical pathway, building the pigment. Now, what's really interesting about this slide is how it visually maps the exact destiny of that heme molecule. Under extravascular hemolysis in the spleen, hemoglobin releases heme, and then a beautifully coordinated biochemical engine just kicks into high gear to start the transformation. Let's break that complex flowchart down into 3 really straightforward steps. First off, the heme molecule separates into iron, which your body smartly salvages for iron stores, and a molecule called protoporphyrin. Then, step 2 is where the main biochemical shift happens. An enzyme called heme oxygenase acts on that protoporphyrin and converts it into something called biliverdin. Finally, in step 3, another enzyme, biliviridin reductase, steps up and converts the biliverdin into bilirubin. And boom, there it is. That's the exact origin of the unconjugated bilirubin that, if there's too much of it, will completely flood the system. Section 4. Types of hemolysis. Intra versus extramuscular breakdown. Now, looking at intravascular hemolysis, you've got to notice the crucial difference here, the location. This happens directly inside the blood vessels. When red blood cells rupture right in the bloodstream, they dump massive amounts of hemoglobin directly into the plasma. Your body tries to manage this mess by using a protein called haptoglobin to bind up the free hemoglobin. But because there's just so much hemoglobin released all at once, those serum haptoglobin levels drop incredibly fast, like off a cliff. And this brilliantly illustrates how doctors actually tell these disruptions apart. Intravascular hemolysis shows a massive drop in serum haptoglobin because it's all being consumed by that free-floating hemoglobin, along with a really big spike in LDH. Extravascular hemolysis, on the flip side, drives a much higher serum bilirubin increase and actual physical splenomegaly because those splenic macrophages are just totally overworked. So the big takeaway here is that while both of these pathways are forms of hemolysis, their clinical fingerprints are totally different based on where they happen. Extravascular breakdown in the spleen is the main driver for severely elevated serum bilirubin levels and an enlarged spleen. Whereas intravascular breakdown, that's characterized by plummeted heptoglobin and all that free hemoglobin just circulating right in the blood. Section 5. Clinical signs and findings. Let's talk about patient presentation. Okay, so when you have that huge buildup of unconjugated bilirubin from extravascular hemolysis, it floods the system. And that triggers very specific downstream effects as your body frantically tries to excrete it. You're going to see increased urobilinogen in the patient's urine and increased stercobilinogen in their feces. Plus, like we just talked about, if it's extravascular, the spleen physically grows. You get moderate splenomegaly simply because the organ is overwhelmed with the sheer workload of breaking down all those cells. So the crucial point is really perfectly captured right here. This dark, high-colored urine sample is a startling visual marker of acute intravascular hemolysis. All that free hemoglobin released into the blood vessels gets filtered out by the kidneys, and that results in hemoglobinuria, which is literally hemoglobin spilling into the urine, turning it a dark red or kind of a tea or cola color. It is a direct macroscopic result of what's going haywire at the molecular level. We've journeyed from downstream clinical markers all the way back to specific enzymes like heme oxygenase and biliverdin reductase. The blueprints are literally inside our cells, just waiting to be utilized. So how might understanding the precise action of these enzymes help us engineer entirely new, targeted treatments for complex liver and blood diseases in the future? It's something to think about. Thanks so much for joining me on this explainer, and I'll catch you next time.